liquid for proper function in liquid disorder. Okay. Oh, there we go. This state is really only in solid state. If we're talking a crystalline state, we're talking everything is organized. It's really just solid. Okay. So we want the disordered state in all cases. That is why the E. coli is changing it. Your body changing its membrane composition also happens. However, you could not undergo those radical changes to have substantial membrane composition changes. Why? You'd get hypothermia. Okay, if we need to change that many cells on you, your core temperature is going to have issues. But is it likely that the membranes will be different if you lived in different regions on Earth? Yeah, it likely is. Okay. What is more characteristic for you to actually alter that's a powerful tool for maintaining membrane fluidity? If you don't, you do use unsaturated and saturated fatty acids, but what would be a bigger player that's more real time for you? What are you thinking? Cholesterol. Cholesterol. Okay. And so when we looked at cholesterol, it has the ability to do what for us? It can stabilize at high temperatures the membranes melting point. It's a planar structure. It gets in there with saturated fatty acids and it helps stabilize it. Okay? At lower temperatures, it also helps intercalate between there and prevents the phospholipids from clustering together and getting too stiff. Okay, so it has both a good and bad. It's sort of the magic bullet here. It's like, oh, protect me if I'm too hot, protect me if I'm too cold. But at extremes, it's an issue. If you take too much cholesterol into your diet, your body will try to excrete a portion of it, but it has feedback loops, and it actually samples all your cells, and it is highly possible that your cells will actually pick up too much cholesterol, okay? That cholesterol, what would it do to your membranes?
Destabilize.
It would destabilize those membranes, which when it goes through a capillary, actually if you have too high a cholesterol, will do what? It will shear open, which will literally rip that cell open and splatter its contents all over inside your vascular tissue, where unsaturated fatty acids and cholesterol, which all are very reactive, react with oxygen and actually oxidize and lead to inflammation and build atherosclerosis plaques. So what is the purpose of HDL? Those who've studied, I got to have high HDL, low LDL. What is the purpose of my HDL?
It takes the excess.
Yeah, go for it.
I don't know. It's described as like a scavenger that takes cholesterol from other tissues and returns it to the liver.
Yeah, in a sense, that's true. It actually mostly comes back through the LDL particles itself. HDL does have the ability to scavenge. What it's really doing is it's turning your cholesterol in your membranes into a cholesterol ester, which allows it to put it in the internal inside of a cell, including your LDL particles, your VLDL particles, your chylomicrons, where it can store it safely. It's not in the membrane. We can store it in the center. That's what you're doing with it. Okay, so good. All right. So let's check a couple other those thoughts that we were working on. Cardiolipin, you drew that, felt good about that. Hopefully able to identify inner and outer leaflet species, recognizing there's a big deal. We talked about this at the end of class, and ultimately the ankyrin and the spectrum. This is on the inside of the cell membrane, right? Mm-hmm. These spectrum are attached to the ankyrin. I kind of described it like it was a barbed wire fence and some fence posts. Are those fatty acids, those phospholipids, moving quickly in the membrane? Yeah, they're fast enough at 1 micron per second that they could traverse the whole outside of a cell. They're really trucking. But they tend to move in certain regions because of this type of architecture. Why would a cell build this type of architecture? What's underneath this that's also being attached? It's other things. So when we jump back to where we're at and we say, what else would I be looking at? Let's see if I can just good. Does cell have architecture? Sure, it's got microtubules that are attached to the outside, microfilaments. What's it using this for? So that it knows when it's transporting things from the outside to the inside and communicating information, where are things located? We generally term our cells to be chaos. We think random. It is, for the eye, looks like that, but to the cell, it knows where pieces are because of what's attached to it. It's essentially got some bit of organization that it's working together, okay? These are not nearly to scale, these are more what you'd see in a cell state or some of those buildings. Okay, so that's cool. We talked about these flip A's, flop A's, scramble A's. We are going to run into the fact that you're going to have to know these even for the upcoming exam. Notice this one's in green. That's probably because I'm honing in on that for the upcoming exam. Flip A's is moving which way?
In.
So the outside in or the inside out?
Outside in.
This is the outside. It's going from the outside to the inside. That's the flip, I. Inside, outside to inside. Flop A's from the inside to outside, the O. Scramble A's, yeah, whatever, it's just going both ways, right? These are called P-type, ABC type, okay? These type of transporters are going to be important as we talk about membranes. And so we're gonna start caring about those. It seems to me like I'm forgetting to talk about something. Oh yes, something that we should have spent some time on the other day. How many of you know what I mean if I talk about a art mosaic or a tile mosaic? What am I talking about? Meg, what do you think? Do you know what it is? What's a mosaic?
I've got a hand here.
What do you got?
Tile mosaic is an art piece consisting of a bunch of small cracked tile pieces.
Yeah, just randomly thrown together, but they make art out of it, right? Okay, so a mosaic is a collection of many different things put together that form some sort of a structure. What is your membrane? A collection of many, many different things thrown together that form a structure. And it's described as fluid. You already understand that because we talked about liquid ordered versus disordered. You talked about maintaining membrane fluidity. You understand that your cell, if it is liquid ordered, is dead, right? Okay, good. But there's a lot of other things in this membrane, right? So integral membrane proteins we've talked about. We've talked about what was on, this is a glycolipid. Oh, that makes sense to us, right? Sugars on lipids. Did we put proteins on the outside that become receptors? Absolutely. One of the ones that we've already talked about were things like hemagglutinin, which was what type of a molecule? It was a protein, like it was actually known as a lectin, right? And the subclass was selectin, right? Okay, so we know those. We have peripheral proteins, we have, so hopefully all this, those sterols being shoved in there. So lots of different things in our fluid mosaic. Yes.
So we talked about how the inner leaflet is different from the outer leaflet, and then if you get the outer leaflet on the outside, or the inner leaflet on the outside, then it goes through apoptosis.
Yeah, okay, let me.
Or can. Why, what I'm asking is, do we flip-flop all the time, or do we only do it when we want to?
Naturally, you are moving things back and forth, but you have enzymes to control it.
Okay.
Even because that rate of crossing is slow, right?
Oh,
On the
okay.
order of about a half a day for one to actually just spontaneously switch sides.
Okay.
So you use enzymes to keep it there. Notice that the one we talked about, the phosphatidylserine, is actually on the outside of the,
Golgi.
on the inside of the
Membrane.
So these are both membranes, but they're on specific side. It's actually a problem all over. If I have it in my mitochondria and it's in the wrong side, it's an issue. The body cares where it is, and so it's a natural signal for cellular apoptosis. It's a natural signal for destroy me, bring phagocytosis along. Go ahead.
So my not to kind of dwell on that topic, seems like moving on, but as far as that point on the study guide about understanding characteristics of the fluid mosaic model, is it just recognizing all those pieces we talked about
It
all
is.
the selectins and all the other particles that are involved in the model?
Yeah,
right?
Kevin's bringing up, he says, that's on the study guide, and it is, it's the whole picture we just talked about, okay? So when you're talking about lipids, when we just talked about saturated versus unsaturated, trans versus cis, you're cluing into why cis are important in a biological setting, and you're understanding that they were created that way by plants, that they're even, but we have enzymes to also re-architecture, bacteria have enzymes to do that. And then you're picking up sterols are part of this picture. You're talking about, oh, there's lots of other things that play into this, but we've got to keep the right membrane fluidity or we'll have problems with our cells falling apart.
Gotcha. Okay.
So that's all part of it, yeah.
And then just to make sure make sure I understand the flip bases and the flop bases. Their job is to, because we would naturally like switch from one side to the other, and their job is to help control that the outside is the outside and the inside is the inside unless we wanted to switch it on purpose.
Yeah,
Okay.
okay, good. So there's the slow right there, right? On the
Yep.
order of a half a day. Okay. Well, you guys, the rates of distances of membranes, all those calculations were up there. If you didn't finish that, please do. Let's go forward. In the same fluid mosaic model, I have built, our cells build rafts. A structure that's thicker and more rigid that will actually be sort of stitched together and will move as a unit. So this lipid raft is high in cholesterol, okay? Contains triply acylated proteins. Wow, what does that mean? That means I got 3 acyl groups stuck off of it, right? We say the word and sometimes we forget we know that. We've got prenylation, we've got things like this cavaline that I'm just going to show you in just a minute when we talk about membrane fusion, okay? But these are all part of our lipid rafts. Notice they're thicker, about 50 versus the 30 nanometers in distance, okay? We often put on the lipid rafts scaffolding proteins. We often put inside of the lipid rafts also proteins that we're putting them all together. Now, we're going to move into a chapter really soon where if this was my reactant and I wanted to do a multi-step reaction, I'm going to hand this to Caleb, he's going to hand it to Lily, and it would hand down. And each time they were touching these, what would we be doing is we're passing this baton. They would be doing their part of the reaction, like an assembly line. This is my part of the reaction, this is your part. And because they're next to each other, it goes very fast. But if the enzymes were all spread out over the cell, that would be problematic. So your body uses what we'll talk about, scaffolding proteins, and uses lipid rafts to accomplish that task of keeping things together so that things go fast.
So it's part of the architecture.
We also want specific curvature. To accomplish that, cavaline, I don't know that I ever say it right, is commonly used. You can see this triply acylated, it's got some cholesterol stuck there. But when I piece these together, I can make a pore. I can make this little cavity inside of it where I can start moving material inside and out. There's also pieces that we use here that are called bar domains. Now, bar domains are actually proteins that are capable of actually creating a specific curvature also of the membrane. So we've got structure of the lipids, and we've got proteins that are helping to create specific curvature, to give it support, to give it angles. Why would we be using that? Because we're trying to accomplish membrane fusion. What was the name of the protein that we learned previously is a fusion protein? Tell your neighbor, I know it.
That's on the virus. Was it the other whatever it's binding to?
Oh, it's salicylic acid.
The other one.
Did you say hemagglutinin? So it wasn't just a binding protein, it was a fusion protein, and that actually is a specific class of enzyme. You'll also see neurotransmitters that we're going to get to that are part of that. Turns out that these are protein-mediated things. So we're trying to get these phospholipid bilayers together, and they're experiencing repulsion, and then we've got to overcome that repulsion, and we're doing that by using proteins to do it. Now, we skipped over that, but here's other examples you're familiar with in biology that do membrane fusion. Budding of the Golgi apparatus, exocytosis, endocytosis, fusion of your endosome and lysosome, or viral infections, fusion of the sperm and the egg, vacuoles, boy, and just separation of actual cells, right? Cell division. There's what you looked at previously with hemagglutinin. It was activated, remember that when it bound, it actually did a conformational change exposing an arginine, which then allowed it to be cleaved into the trimer that it was, into actually dimers within that of each of those individuals. And then it opened up. Massive change once that cleavage occurred that actually caused this weaving together where it's actually pulling these into fusion. So the one we're going to talk about that the text brings up is something known as, a neurotransmitter release. Now, I happen to know the biology faculty. It was so fun to go to a discussion about one of their big accomplishments in teaching a biology class about this very thing, and how they drilled it into you over and over and over again. And then they covered it in like 2 seconds and said, see if you can teach somebody else. And then I'm sitting next to Brother Kelson as well, and he's just like, how did you know all of that? In fact, you expounded on it. And I'm like, this is basic stuff, right? This is basic. We're covering this biology piece not because of the biology, we're covering it because we're interested in the biochemistry, right? It's the same theme we've been going on all over again. How do molecules interact with each other? How do they change shapes? How does these inanimate objects make life? That is the beauty of the biochemistry. In biology, you're looking at the life and we're describing what it's doing and its trends and patterns and names of everything. In biochemistry, we're looking at why. How do I take it from the dust of the earth to a living cell? To me, this is the beauty of it, but it's all one subject. So you should know what a snare, a snap receptor is, D snares, vesicle snares, T snares to the target snares. What are these things talking about? We're talking about a neurotransmitter being released as I'm going along neurons, and gosh, I'm going to do what? I receive my signal. Let's assume that I got acetylcholine and just activated, open the pore, and in rushes some sodium and calcium, which does what? It depolarizes the cell and allows for action potential along the neuron and then that continues to cascade until it opens what? The next calcium channel, which when it runs in, the calcium, a very common one that when it comes in releases vesicles. These vesicles right here, acetylcholine, which then come to the outside of the membrane, and what do they have to do? Exocytosis. How is that done? Yeah, so we use this approach where we use the membrane fusion and the neurotransmitters filled, they zipper together, they actually weave together. How could they do that? Tell somebody else some guess that you might have. How could these proteins actually start weaving together?
That was like, that's what I was about to say.
It's actually
Probably.
Generates that.
no, actually, could be anything. Small down. Maybe it's all. Definitely not scary.
That's an okay chemistry.
It's always amino acids.
Well, if you have those
might be
proteins,
scary.
right? The VCR and
This is like the OChem, like
So,
golden child age.
so
Yeah, that's totally.
in your discussion,
And
did
but
you
they're-
think
they're
about the
doing-
fact that
they're
it was
doing-
calcium
gonna
that
cause
triggered
fusion to happen.
that action? So the calcium's an ion that must be disrupting a salt bridge, right? Which is then allowing that to open up, and then new interactions are forming, which are allowing this action to happen. Interactions between proteins that are electrostatic, hydrogen bonding events, and this fusion then occurs, allowing for the release. Okay? Beautiful. Wow. Vesicles are snares with arginine, Q snares with glutamine. Oh, so these were part of D and D snare, T snare. Okay, 4 alpha helices coming together. Okay, curvature with this. There's this gee whiz fact. So NSF, ethyl maleimide sensitive fusion factor, so NSF, acts as a disassembly of the snare complex when the fusion is complete. Well, here's something that would be interesting for somebody that understands botulism, because why? I keep bees. What's a common reason you don't feed a child honey, raw honey?
Botulism poisoning.
Botulism. Well, you better be careful. So a colostrum, a botulinum toxin, whether you cook it or not, is still there. Oh, darn it. It contains a protease that actually is capable of doing what? Cleaving the snare bonds, preventing the fusion for the neurons, and this results in paralysis and death. You just figured out why botulism is bad for you. The protease, messing up your snares. Okay.
Wow.
That's our fusion protein discussion. Huge, right? Life-changing. Maybe not. Delta G. When we talked about delta G before, some of you would have thought about concentration of one versus another, your negative RT-LN of that. But you might have forgotten that there's also a charge species over here. I've got to take into that as well. How do I handle these polar molecules? How do I handle the charge species as well? So there are specific calculations that you can go into that talk about this effect. Now we're going to gloss over this at high speed, and then if you're in second semester, we go through it in gory detail. Osmosis, right? What's it got? If I got a semi-permeable membrane, it will eventually reach equilibrium. What would happen to the charge? It will also equalize given the opportunity, right? So, molecules come. Gosh, I've got to get across the membrane. What does this look like?
Well, it's a lower energy of activation.
So, within less than 3 years ago, they introduced a new enzyme class of the 7 enzyme classes, which one is this? Transporters. Huh. It's lowering the activation energy required to cross the membrane. That makes perfect sense. So we always knew about the transporters. We always knew what they were doing. We just decided to call them enzymes. Okay, so here's our transporters. We've got to know some things about transporters, and let's see if we can make sense of it. This is like so cool because this is like when I learned in high school, things like simple diffusion, facilitated diffusion. What's the difference? One requires a protein that assists in it. Simple diffusion, just getting in. Do we have molecules that can just cross membranes without? We even talked about drugs that could do that. That's cool. Then we have something called if I'm talking about facilitated diffusion or simple diffusion, which way does it go?
High to low.
High concentration to low concentration every time. Okay, but if I'm talking about primary active transport, what are we talking about in concentration?
Low to
I'm spending ATP to go against the gradient. So I'm using the protein's ability to adhere to a molecule, grab it, and then I'm pushing it uphill in the concentration. ATP is used in this instance. Then we talk about ion, here where I'm moving them in something that we'll talk about. This is a syn, but this is, we'll talk about syn and anti-transport in a second. Secondary active transport here, what's happened is I built a gradient with my active transporter, and then I use that gradient difference to go high to low, and I carry along a molecule that then is going against the gradient, but another molecule is going with the gradient and it's bringing along for cargo.
Okay.
And so that's a secondary active transport, yes.
Sorry, to make sure
We're going to talk about it more.
we're talking more into detail.
Yeah.
Okay, I can.
This is
hold
just
on
the
to that.
overview slide.
Okay.
An ion channel, just open the channel and ions are going to move based on their
Electrochemical gradient.
electrochemical gradient. Okay, and an ionospore become interesting molecules. But this is a molecule that's capable of actually engulfing an ion, encasing it inside with the charge, hiding the charge, and essentially doing simple diffusion as a result.
Wow.
Okay. So 3 classes of transport: uniport, symport, antiport. Did that have green on it? Did that have green on it?
Oh, yeah.
That looks like way easy questions I'd hate for you to miss on. Because this is low proof. But this is where we're talking about an antiport, one going in way one goes out, symport moving together, uniport just going with the gradient one direction, right? Ion channels, yep. So we're going to do a case study on our GLUT transporters. There are more GLUT transporters. There's 12 GLUT transporters. We're only interested in a couple of them here. GLUT1, GLUT2, GLUT3, GLUT4. The GLUT1, typically found in erythrocytes, meaning your red blood cells, right? Your GLUT2 transporter found in your liver, your epithelial cells. Okay, facilitated diffusion, both of these. GLUT3 found in your neuronal cells, or GLUT4 in the muscle and adipose tissue, insulin-dependent facilitated diffusion. So here comes a GLUT4 transporter that you just saw. Okay, so in muscle, fat, heart. Insulin receives a message. We'll talk about this in the next chapter. So this is a tyrosine kinase. It's actually phosphorylating something. It's causing in that event a reaction where the GLUT transporters are moved from this vesicle to the membrane. Insulin was a signal that said I was
Ready.
My blood is filled with sugar and I want to get it inside of my cell because I need some energy. And if I leave it out in my vascular tissue, what would happen? Tell somebody else you remember what happens if I leave raw sugar out in my vascular tissue.
It'll start.
Bad things.
Bad things. Okay, that's right.
You don't want it.
He talked about advanced glycation end products. He talked about non-enzymatic glycation. That's what we're reviewing. Why am I doing this? I'm trying to interweave the recollection. You have a final that's coming really right at the same heels that this one's coming, right? So the GLUT transporters come to the surface, and then they bring in the sugar. How does it do that? This is that GLUT transporter, okay? So, high to low concentration. Here I'm looking at this GLUT1 transporter. This is interesting. Polar charged hydrophobic. Now hydrophobic inside my membrane makes perfect sense to me. But I've got all of these polar charged right here. What are these circles?
The hydroxyls?
These are alpha helices. How many alpha helices? 1, 2, 3, 4 alpha helices. What's this charge in here? This is polar right here. These are non-polar. It's made a protein where it took all of this and did what?
Made a little
alpha
basket.
helices that had a pore right down the center of it, and the pore was polar.
Yeah.
Wow. Huh. That's clever. Somebody was thinking how to do that. I'm sure it was a good accident. Yeah, right. So when that GLUT transporter gets there, this is a more intricate look at the very same thing you were looking at. Remember that it's these helices, but you're having a molecule where the glucose signaled is then coming in. Once it's inside of that trap pore, it's actually doing what? It's got a conformational change when the glucose gets there. So it's not exactly a hole. It's actually doing a change. That conformational change, and it rocks back and forth to move that molecule across. Glucose transporters, then what would they have? Oh, this looks like horrifying. I recognize something like this. A Vmax. Someone have Vmax. Oh my goodness.
I swear I would never go back.
K's instead of KM for transporters. Oh my goodness. Lineweaver Burke stuff. Oh. Do you remember all this? Good. I'm so glad you learned something in this class. Wow.
Not this stuff.
Okay. Okay, we have other GLUT transporters. Okay, this is our GLUT2, okay, in our epithelial cells, literally lining the stomach. Okay, so here we are. We have these glucose out here in the stomach, and I've got it, and I'm going to move it across. Okay? Now, in this instance, the first thing that's happening is let's come over here, let's do some active transport with something known as the sodium-potassium pump. And we'll study it more. Which way is the potassium going? It's going into the cell. Which way is the sodium going?
Out.
And I'm using ATP to accomplish this feat. I'm only moving 2 charges and 3 charges. Wow, the charges off isn't the same, and the concentrations aren't the same. I just build up. Do you know how much energy we spend doing that? Like 1/4 of your entire caloric intake is spent doing that alone. You want to sit like a couch potato? You're still burning energy doing this. Great. Okay? So I build this up, and then over here, looking at what's going on, the glucose is going to come in. How come the glucose is going in at all? This is a symport, right? And it's bringing the glucose into the cell, okay? High concentration, okay?
And then
in doing so, it brought the sodium. How could the sodium go in? Concentration gradient, I pumped all the sodium out. It wants to pull it in. It's easy, just like that neurotransmitter, right? And then the glucose needs to go out. This is just a uniport, but it's going from high concentration to low concentration, which meant this was doing what? This was high or low concentration compared to inside the cell. Yeah, lower, and it went to a higher concentration. That's why we use that facilitated fusion. So that's technically a secondary active transport.
So we see these proteins on like rafts working together, or
That's a great question. So this one doesn't require a lipid raft to do that purpose.
okay.
But if I was talking about something like inside the mitochondria, the electron transport chain, where I've got complex 1, 2, 3, 4, they're all going to be built together, right? If I want to do the whole citric acid cycle, just to be honest, guess what? You're not going to keep all of the things just randomly floating around. You're going to put all the enzymes together. And we purified them, and we know they come together. Yeah.
Can you have a secondary active transport be uniport or symport?
Yeah, you could. Most of the time they're symport.
Is that like
Because what I did was I built up,
Created gradient.
I
for this-
created a gradient that actually allowed for the one to pull somebody else in the process. Okay. Yeah, we'll take some.
So, does that mean that a secondary gradient or a secondary active transport is often facilitated by like a primary active transport
They
somewhere
always
else? Okay.
They always are. Yep.
Okay.
Good. Okay. So there's no review in this section at all. You would know nothing about something known as carbonic anhydrase, right? Wrong. Tell me about carbonic anhydrase. Where did you see that enzyme before?
Yeah, blood.
With
It changes.
That
was the pH.
Oh
it turns CO2 into carbonic acid.
I had a lot of CO2 in the tissues. Why did I have all that CO2? I was doing metabolism, and that CO2 was then lowering the pH. How did it lower the pH?
It's not hemoglobin.
Respiring
We
tissues
were
pulled the
talking about
CO2 in
hemoglobin.
and made it into
That was
carbonic acid, which did what to my hemoglobin? Moved it from R state to T state. How did it do it? What did it activate? On the outside, some amino acid. What some amino acid?
It was
It was a histidine. You're right. But there were 4 histidines we were talking
It was the
about.
last. It was the
That's
last one.
frightening. Now I'm really messed up. With distal, proximal, there was this histidine on the outside surface of histidine. Was it 57 or no? Wait, that one might have been for our enzyme kinetics. What was it?
133.
HC3? Yeah.
Oh yeah, that one.
Right. Easier for me to remember that way instead of the 148
Never could forget that.
or 145. And that histidine, when it became protonated, caused a
Salt bridge.
salt bridge between a.
Arginate.
With
Lysine.
FP1.
If it's positively charged now, what would it have to be interacting with?
It's like
A negative.
Aspartate, good. Yep, and then it connects, moving the alpha helix position, which then affected the distal or proximal.
Proximal.
The proximal histidine, which was attached to a porphyrin ring, which then folded, which then caused the kicking alpha. This is what you need to know.
Stop action.
Goodness.
Boys.
If it hasn't crossed your mind, if you're not studying for the final at the same time you're studying for this exam,
Bad news bears.
you won't have time. Start budgeting. As you're filling out the new study guide, realize you will only need to know it for the test because you'll already have studied for the final part of this portion. Which means you've got, instead of 5 units, you only have to focus on reviewing 4. And it would be crazy to think you were going to read all those chapters. It would be horrifying if you just took blank study guides. But if you grabbed a blank study guide and you start reading through it and you're like, I remember that. Oh, I don't remember that. Then you could pick up your study guide and you'd figure out what
the answer was.
What the answer was, without having to go through the goriness of the book. And your cliff notes would probably bring most of it back to your memory, unless you never learned it in the first place.
Dang.
Okay, so carbonic anhydrase works not only in the tissues, but it went in reverse the other way around. What was it doing here? What is this called? Symport.
That's
Antiport. Oh, it's an antiport. This carbonic acid going out, white chloride's going in. Their charge is the same. That's keeping electronically neutral. Oh, that's pretty cool. Okay. Simple diffusion, antiport facilitated fusion. Yep. Awesome. Okay. There's that primary active transport, here's secondary active transport. We already talked about it. Does everyone understand the difference between the two? Yes, the primary active transport is always used when I'm using that secondary active transport. Meaning I needed to build up a gradient, so I'm going to use the primary active transport with ABP to create the gradient difference, but then I will do something like a antiport or symport usually. Okay. So those were types of transport. Now we're drilling into the transporter names. It gets worse, yes. So there's only a few that you need to know in the next 12 minutes. An ABC transporter is one of them. An ABC transporter works like this. It costs 1 ATP, a flopase that we just talked about, which was going from what? Inside out or outside in?
outside.
Glad you got it, inside to the outside. Okay, it's a nice example of an ABC transporter. What is it doing? By the way, this is not a flopase. This is just, that is one, okay? The substrate binds. In binding, it actually goes with a nucleotide binding domain. ATP is being used because this is an active transporter, and it binds the nucleotide binding domain to ATP, which actually causes it to do a conformational change with the substrate and the ATP in position. That's how our active transporter works as an ABC transporter. An ATP binding cassette is the ABC.
Is the sodium potassium pump also an ABC transporter?
Let's look. And the answer is no. It's a P-type. Okay? So this one, this chloride channel, acts like an ABC transporter. In fact, it has a nucleotide binding domain. So I have just showed you 2 examples of ABC transport. Okay? And they function the same. Essentially, once bound, you change and bind the substrate and the ABC and the nucleotide, the ATP, then it will change shape and allow entry of the protein. The next one is the P-type. So your question was, what is the sodium potassium pump, and it's sitting right here. The 2 we're going to look at is ATP, calcium ATPase, or known as the circuit pump, okay, and sodium potassium pump. There are others that are involved, okay. This one's slightly different. It has a nucleotide binding domain and a phosphoryl domain and an actuator domain. So it has 3 pieces as we transport across. Now, I'm going to show you, so we're going to look at this in detail with the calcium circuit pump in just a second. But first, I'm going to do a little background on the calcium circuit pump. Calcium ATPase, known as a P-type transporter. ABC, now P-type. Has it ever crossed your mind what happens when I put a calcium with a carbonate? This was in 105. It was something with solubility rules.
Forms a solid.
What happens with calcium and carbonate?
I mean, does it form a solid?
It forms a solid. In fact, seashells are made out of it. Limestone is made out of it. I'm making rock if I have calcium and carbonate next to each other. Wait a second. Every cell is doing active metabolism, and every cell has
Calciums and carbonates.
calcium and carbonates. I better be in control of this, or I'm going to be in trouble. That's right. Okay? We have to keep the calcium in the right place and a smaller concentration of it. Okay. or else we'll get death, okay? So, yeah. I'm gonna move my calcium around this way. I'm gonna use a p-type transporter. Here's the detailed goal. ATP comes in. What's the charge on ATP?
It's negative.
Negative. Got three, it's a whole bunch of negative charge, right? So it actually, when binding to the nucleotide binding domain, it actually pushes it into the phosphoryl domain. It actually undergoes, at this point, a hydrolysis. We're not showing you all the details. In hydrolysis, I'm actually got a magnesium. Wonder where that came from. Oh yeah, it was hanging on ATP. And some phosphate, a phosphoryl group, and ADP. Once this is here, what did it do to the channel where the calcium came in?
Closed the channel.
Closed it, but it didn't do it here. It was open in this position. Now it's closed. So the channel's now closed, and then in position 3, what happens? The phosphoryl group, the ADP leaves, which moves the actuator domain. In moving the actuator domain, it actually opens the pore on the other side so that the calcium isn't just closed here, it's now leaving. We're talking about going uphill. If I have a pump and I want to pump something uphill, I need to close a door from the one so I can push it uphill. That's really what we just did. And then I'm going to release the magnesium and inorganic phosphate, which will actually reopen it so we're ready to reset. Okay? Now you can look at vanadate and understand why vanadate might be a problem too. Coincidentally, another p-type transporter would be flipase. So p-type was 3, sodium potassium pump, calcium circa, and flipase. Go ahead.
I just wanted to get an example of this. Like, with this calcium, like isocyl, is that kind of weird? Is that neurotransmitter? Not neurotransmitter, but the neuron binding, or?
So how did I get the calcium so it would rush in for that neurotransmitter when we just looked at the acetylcholine? I used the calcium circuit pump. I pumped it uphill, and that's ready for action potential. Then I can use.
Yeah. Okay.
Okay.
Yeah.
All right. So that's really what we're doing here. This right here is the sodium potassium pump. You should know how many sodiums to how many potassiums, because otherwise you would be unintelligent that you were graduating from a biochemistry class. Okay, that makes sense. Perfect. And it uses ATP. You don't need to know the concentrations. Yes, 25% of your total metabolism. Okay. The next type of transporter, an F-type. Oh, one more. Okay, ready? Ooh, this one's cool. We go into gory detail later on this, but this is called the ATP synthase. It's the end of the electron transport chain. This uses protons to actually create ATP. But we also use it the other way around. We use ATP to pump protons. That's how I could get a vacuole to do that. But if I want to use the protons to make ATP, I can do that. So ATP synthase is ultimately a reverse direction of an ATP F-type. Wait a second. Well, then what would I use it? Yeah, making my vacuoles acidic. Pump some protons in so I can break down things. Wow, that's a good idea. What else could I do? Oh, I was going to show you another one. Where do we think the ATP synthase came from? Turns out that I use protons in a bacteria and ATP, and I pump it through so I can actually turn a rotor. It's called a flagella.
Oh.
And the actual protein homology, almost identical. Whoa, that's confusing. It makes sense though when you start understanding how it was designed. Okay. There's an ionosphere right there, just encasing the potassium go out, go across. Last 2 seconds. These ones are still show up on the study guide. I don't spend a long time on them, but they're amazing, okay? So, F-type, P-type, ABC, and ionosphere, which we just showed you was a way of encasing the ion by making it polar inside and non-polar on the outside. This right here, though, is a channel for active transport. It might not have crossed your mind, but potassium, the sodium potassium pump we just talked about, or moving a potassium around, how do I know the difference if they're both a +1 between a potassium and a sodium?
Size?
Size, sure. But can I really distinguish that type of small size?
No.
Answer is, it's actually the waters of hydration size that actually is the perfect fit. The waters of hydration actually keep the wrong one out and the right one in because the size of the actual ion is perfect that I can replace the waters of hydration with the protein charge groups as it goes through the pore. I'm going to strip a water of hydration off, and it can only come off because I want to keep the ion in solution if I have something that can substitute with the exact hydrogen bonding, which is set with the protein that's matched to that. So that's how we do that. We can actually distinguish between sodium and potassium. There's their different size at a 10 to the 4th specificity. To a degree that we can go 10 to 8 ions per second, which is as fast as you can possibly go. Wow, that's incredible. You want to see one faster? An aquaporin. Water. And it can move water and not move hydronium. And it's designed to not allow proton hopping, which is where the proton could actually move on the hydronium by going and binding to a water, becoming a hydronium, and then switch another proton come off of that one and just move it along the water without actually physically moving the whole hydronium. But this one's capable of doing it. What is it doing? Electrostatic repulsion, so it keeps the charge groups away. Size restriction, water orientation to flip it so that it can't just do, it's all designed in there. It runs at 10 to the 9th per second. You want to know aquaporin? Your salivary glands. Wait, that's fast. Yep. Those are aquaporins. They're pretty cool. Okay? All right, that was chapter 11. Awesome. Yeah, you could go do some awesome math. That'd be cool. Yep, that'd be cool. Yep. That's what we were learning today. Wait a second. Brother Bowden, that went so fast. Did I learn what I was supposed to? Take a look at the study guide, make sure that you're up on things, and we'll be right. Oh, let me pull the study guide up. Sorry, guys.
Thank you.
Bye, John.
Chapter 12, Friday. And we never slowed down, right? You're like waiting for it to slow down.
Going.
We can't. We haven't got the time.
Oh my goodness. This should be a 4-day class.